Good afternoon, everyone, and welcome to today's webinar, which is called The Risk of Substandard Counterfeited Anti-Cancer Medications in Clinical Care Settings and Private Pharmacies in 4 African countries. So this session builds on growing evidence that the scale of unsafe medicines such as substandard or falsified medical products across Africa is rising. And today we will have a particular focus on a critical but often overlooked issue, the underdetection of poor quality medicines for treating cancer across the continent. And the key message is simply this, that these risks are not isolated incidents. They are deeply rooted in fragile and fragmented pharmaceutical supply chains where weak market surveillance and detection systems are allowing harmful products to reach patients without being picked up. And so today, we are going to use a multi-country real-world evidence analysis and case-based learning to provide a practical understanding of how medicines quality failures are occurring in the African context using a number of countries that you will hear shortly, and how health systems can respond more effectively. And so our session today will be presenting an analysis led by Prof. Maria Lieberman in a number of African countries represented here by the panelists. And I will introduce all of the panelists shortly. But first, in this webinar series, Building Africa's Frontline Defenses Against Unsafe Medicines, we have this tradition that before the main speakers come on, we do a brief recap of the previous webinar in the Frontline Defenses series. And it allows people to join and get time to get home and join the webinar before the main session. And so our last session explored the risk of nitrosamine contamination of essential medicines. And it was presented by Dr. Partha Mukherjee, who is the Executive Director of Analytical Chemistry at Eli Lilly and Company, the big pharma US company. And that webinar examined a case study on nitrosamine risk assessment approaches taken through the European Medicines Agency and the US Food and Drug Administration. You can actually watch that webinar on our YouTube channel. We have shared the link in the chat here. Lydia can reshare it as well, so you can see it. And also, it goes for the first webinar that we had from Dr. Martins Iliomade, who is the Director of Investigations and Enforcement at the Nigerian Food and Drug Administration, the NAFDAC agency. And so you can watch those. They are recorded and they are available for you to see. But for now, Dr. Kimo Makinen, the co-founder of Medicines for Africa, will provide key insights from the nitrosamine contamination webinar, the last one. And with that, I am going to hand over to Kimo for the recap. Kimo, you've got the floor.
Thank you so much. Can you hear me well?
We can hear you very well.
Excellent. Yes. Tell me also if the slides are going into presentation mode, because I'm trying to do it here so I can get going.
Yes, they're in presentation mode, yes.
Excellent, yes. So I won't take much of your time. I'm just going to run in a few minutes over what I took as the key learnings from the previous webinar. So in this way, I'm like an opening act to the main act of tonight, if you like. So nitrosamines are small molecules with a nitroso group that are potentially carcinogenic in humans. You see here a little chemistry. So whenever an amine interacts and reacts with the nitrosating agents like sodium nitrate under specific conditions like acidity, you can have a formation of nitrosamines. They are highly carcinogenic and mutagenic in rodents and in animal models, and they are considered probable mutagens and carcinogens in humans. They're part of the cohort of concern in ICH M7, which means that they need to be monitored, and regulators require that they are stringently monitored in medicines. You can get exposed to nitrosamines from various sources, not just from medicines, but the main source is actually the environment. For example, certain drinking water that contains nitrate can react with disinfectants to create nitrosamines. Cured meat, certain fermented maize drinks, tobacco, smoke can all contain nitrosamines, and that actually accounts for 75% of the total exposure of nitrosamines in our daily lives. But pharmaceuticals are also a potential source of nitrosamines. They have a lower absolute contribution to the daily load, but they're problematic, especially if you have long duration treatments, like for diabetes, for example, that you have to take for a long time. And because the levels of nitrosamines in pharmaceuticals can be controlled, they must be controlled. And that's what regulators are asking us to do. Nitrosamines are, broadly divided into two categories. You have the small standard nitrosamines, which are volatile compounds that are unintentional byproducts in pharmaceuticals, food, and environment. They are completely separate from the drug molecule. They are not related structurally to it in any way. First case was detected in 2018 when valsartan, which is an anti-blood pressure medication, was found to contain NDMA, and it was pulled from the market. And certain drugs, classes of drugs like antihypertensive, antidiabetics, certain antibiotics, so on and so forth, are especially at high risk of contamination with nitrosamines. NRAs are very aware of the nitrosamine risk, and they have set clear limits for these kinds of compounds in drugs. For example, NDMA is considered allowed at 96 nanograms maximum per day exposure, and these are very low levels of nitrosamines, or levels that are allowed to be found in medicines. Standard analytical methods are available to detect these types of small standard nitrosamines. It doesn't mean that they are necessarily accessible, but at least they have been developed and they are available for this class of nitrosamines. The other class, the second class of nitrosamines, is called nitrosamine drug substance related impurities, or NDSRIs. These are structurally similar and derived from the APIs, and they form when the API that contains secondary or tertiary amine reacts with nitrosating agents like sodium nitrate during manufacture, storage, or packaging. They are always named as N-nitroso and then the name of the API. For example, here on the left, you see tolbutamide reacting with sodium nitrate and becoming N-nitrosotolbutamide. The first case of these NDSRIs was detected in 2021 when the smoking cessation product varenicline was found to contain N-nitrosovarenicline. The acceptable intake really depends on the API, and it has to be determined case by case. So there is no set limit for NDSRIs. It depends on which one we are talking about. And also derived from that, there are no standard methods available to detect them either. If you have a new NDSRI that you think you might find in your product, you have to develop a new method. And that's exactly what our previous webinar covered by Dr. Partha when he was describing the specific case study that his company was involved in. So check it out if you're interested in this kind of method development. Now, the actual number of nitrosamines that we know of has been expanding over the years. In 2018, we only knew of one, NDMA, but now we know many. We know many more of these small standard nitrosamines, and also we're learning all the time more about these new NDSRIs as well. They can actually be, arise in so many different parts of the medicine manufacturing process. They can come from the excipients. They come from the utilities. If the water, for example, contains nitrates, APIs, can convert into these NDSRIs as well. Manufacturing process can, introduce nitrosamines, and even packaging can react to create nitrosamines, right? So therefore, regulatory dossiers these days must show that you're controlling all of these areas, during medicines manufacture. And you have to perform a thorough nitrosamine risk assessment. So this is now a mandatory component of a drug registration dossier, and it's divided into these different parts in here. Number one part of this risk assessment is the chemistry. Does this medicine have the potential to form nitrosamines? Does it have the kind of chemical groups, the kind of amines that could potentially lead to nitrosamine formation? Number two is toxicology. Is this potential nitrosamine predicted to be mutagenic and carcinogenic in humans? And usually this is a structure-based prediction. And if it is, then what would be the acceptable intake level per day? Number three, do you have the analytical method available to detect these nitrosamine products? And the detection has to be done at 10% of acceptable intake level at minimum. Number four, in vivo metabolic bioactivation. Can this nitrosamine be converted in a human body to an active mutagen? Number five of the risk assessment is the clinical risk. What is the dose and duration of this medical treatment? Is it taken for a long time? Is the dose high? Because, the longer the duration of the treatment, the higher the dose, the higher the risk as well. And number six is the regulatory risk assessment. Will this dossier meet the requirements of the NRAs? So how do you detect nitrosamines? Well, you need top of the range equipment to do this. You need to have highly sensitive detection, usually at parts per billion level of sensitivity is what we're shooting for here. And this calls for expensive instruments like liquid and gas chromatography, followed by mass spec. These are not widely available. You do have them in certain places, but there's definitely a gap that needs to be filled. These same kinds of instruments like, GC and LCMS are used to detect both small standard nitrosamines and also NDSRIs. But for these small standard nitrosamines, the methods have already been developed and they are available. Whereas for these NDSRIs, if you have a new one, you have to make a new method. Right? And that in itself can be a lot of work, which is what our previous webinar speaker described. So finally, to mitigate against the threat of nitrosamines, what we should always do is buy from quality sources. We should know also the risky products and conditions, that can lead to formation of nitrosamines during manufacture as well as during storage. And we should test periodically. So market authorization holders are actually responsible to ensuring that each of their batches of finished products is of satisfactory quality, in this case, in terms of nitrosamines, showing the purity of raw materials used, that they have controlled all the steps of manufacture for nitrosamine risks, and they must be able to produce a full nitrosamine risk assessment if you request it. In countries itself, it's important to perform second line testing to verify levels of nitrosamine, because even though the market authorization holder has a dossier, and has their methods, the methods may not be perfect. So it may be a good idea to check for yourself. And also because nitrosamines can form during storage, especially in hot climates, they can form. So it's a good idea to check periodically, giving priority to those medicines that are most likely at risk, because some classes are riskier than others, and also those that bring the highest impact on patient safety. So if you want to deep dive into this topic, check out the webinar by Dr. Partha Mukherjee. It's on our YouTube channel, and it goes into great level of detail on this topic. Thanks very much. I'm handing back to you, Lenias, so you can then kick off the main webinar of this evening. Thanks.
Thanks a lot, Kimo. And everyone, we just shared Dr. Partha's webinar recording in the chat, so you can take note of that if you want to follow it. Now, let's go to today's webinar, which is, again, just to remind people that on the risk of substandard and counterfeited anti-cancer medications in clinical care settings and private pharmacies in 4 African countries. So very briefly, this session is going to feature a structured presentation by a panel of experts, and then it will be followed by a discussion, which will provide an opportunity to discuss this real-world evidence and the wider implications that it has for patient safety. I know people love to discuss afterwards, so I'm really looking forward to that. So today we are truly privileged to welcome a very exceptional multidisciplinary panel of experts who are working at the intersection of cancer care, medicines, quality surveillance, and pharmaceutical sciences. Today's discussion really is going to be led by a team of experts that is working in 4 African countries, that is Ethiopia, Malawi, Kenya, and Cameroon. And the publication they're going to present was led by Professor Maria Lieberman, who was the leading author. And she is a cancer research specialist whose work focuses on innovative approaches to cancer detection and medicines quality monitoring. On the panel today, we have Dr. Atalay Mulu, who is an assistant professor at Addis Ababa University, and he brings expertise in oncology pharmacy and cancer patient care. We are also delighted to welcome Ibrahim Chikowe, who's also an associate professor for medicinal chemistry and toxicology at Kamuzu University of Health Sciences in Malawi. And his research focuses on pharmaceutical quality, toxicology, and public health. Joining us from Cameroon, but is in Uganda right now, is Dr. Yawuba Saidu, a physician scientist with expertise in global health, clinical trials, and health system strengthening in resource-limited settings. Lastly, but not least, we have Dr. Ayanu Ashene from Addis Ababa University as well, and from the World Bank Center of Excellence for Therapeutics and Drug Discovery. And his work centers on pharmaceutical sciences and drug quality assurance. So together, these panelists are going to share with us findings from their real-world work from across 4 African countries, and they're going to highlight substandard and falsified anti-cancer medicines, how they enter the clinical care and pharmacy settings, the risks they pose to patients, and the practical actions needed to improve surveillance, detection, and better protect patients across the continent. And they are also going to talk to us about some of the cost-effective solutions that are coming up. So we'll have a presentation segment that will run for about 45 minutes or so, after which we can have an interactive panel discussion with audience questions. And so without much further ado, Yawuba, Atalay, Ibrahim, INL, and the rest of the team, good afternoon and welcome. The floor is all yours, and so we can begin. We need to get your presentation up. Kimo, the team suggested that we control the slides. Are you able to just continue since you were already on this track?
Yes, just give me a second. I'm just going to put it up.
Thank you.
All right, we should be ready to go. Just please tell me when I'm to change the slide and I'll control them from here.
It's in full presentation mode. It's ready to go. So speakers, over to you. Your slides are up. I don't know if you can see them.
Thank you very much for this opportunity to share the important information about the cancer quality in sub-Saharan Africa. In this study, we collected samples from 4 countries, which include Kenya, Ethiopia, Cameroon, and Malawi. And so far, we have tested 394 lots since 2020 to 2026. And out of these samples, 1 in 6 failed quality assay HPLC, which were confirmed by the HPLC. Next. Yeah, so this team is led by Prof. Maria Lieberman, who is an analytical chemist. And then she develops technologies for low resource settings. And she represents the team of researchers from 5 countries. And she began studying the quality of oncology medicines in sub-Saharan Africa around 2017, where she was working with the Prof. Ian Ashnef at Addis Ababa University. In 2022, the project expanded with a grant from NIH SCT program. First of all, why do we need to study this class of compounds? And then the thing is, pharmaceuticals are highly regulated products, but each country is responsible for implementing and enforcing the regulations. And the countries as well vary greatly in their capacity to do this task. Next slide, please. Yeah, so the goal of the presentation is in 3 folds. The first one is to define the problem. And then here we'll describe the scale and the clinical impact of substandard and falsified medicines, particularly in sub-Saharan Africa. And then the second one will be introduction of the solution, which in this case will describe the role of the chemopad, which is a point-of-care technology whose aim is safeguarding cancer patients from substandard and falsified anti-cancer medicines. Then we'll evaluate the evidence, which will involve analyzing both the successes and limitations of chemopad and the related approaches based on the real-world field experience that we have had. So in this case, the WHO classifies national regulatory systems using the global benchmarking tool. The regulatory systems at maturity levels 1 and 2 cannot reliably perform some essential regulatory functions. For example, a country that does not have a pharmaceutical analysis lab cannot assess the quality of medicines. At maturity level 3, the regulatory systems have a reasonable ability to perform most regulatory activities, while at maturity level 4, the regulatory system can perform all the needed functions, and it is a global leader or model. Now, out of the 54 countries in Africa, 45 are at the maturity levels 1 and 2, while only 9 countries are at the level 3. And if at 4, so far, there's no country that can perform these kinds of regulatory activities. So this photo demonstrates a worst case scenario for the manufacture of a medical product. This diagram represents a setup that was identified somewhere, showing how a fake Viagra was being manufactured in a completely unregulated site. The ingredients for these blue pills are being stored and mixed in a data apartment. And then in this case, who knows what is actually being put into this pharmaceutical cement, which cement mixer, or whether it is safe for the patients. However, the pills are pressed and packaged. The product can look exactly like a legitimately manufactured product, yet it is a falsified product. Products that don't meet those standards are called substandard, while the products that deliberately make false claims about their identity composition, or the source are called falsified. It is relatively easy to go into the lab and identify products that fail standards, but it is much harder to pin down false claims. The responsibility for ensuring the quality of products in a particular country's markets falls upon the country's regulatory authority, and as we have seen, countries vary widely in their capacity to execute these regulatory tasks. One consequence is the pervasive presence of substandard and falsified pharmaceuticals in low and middle-income countries that do not have adequate regulatory capacity. So from here, I'll ask my colleagues to continue the presentation.
Yeah, this is an example of just a review paper done by Professor Sachi, which estimates the prevalence of SF products, substandard and falsified medicinal products, at 40%. This is also substituted by WHO just in 2017. 1 out of 10 products in circulation in low and middle income countries are substandard and falsified. Next slide, please. So how the regulatory agencies catch this kind of products, substandard and falsified products, is by doing regulatory assessment that includes registration of drugs, import-export controls, just they oversee manufacturing, assess CGMP compliance, good dispensing practice, and post-marketing surveillance, as well as enforcement of the regulatory activities. Post-marketing surveillance is best because it just assesses the actual scenario, where the drugs are stored, the supply chain, and how the patient uses. So that is highly encouraged. Next slide, please. So in global surveillance system, many fake drugs can be just caught off. For instance, this is Ponsati, anti-cancer drug, but it was found to be only paracetamol, and so on. So the surveillance systems catch many of these, and we don't know a patient takes paracetamol rather than the required anti-cancer drugs. What it affects the patient, just we can imagine. Next slide, please. So there are many chemo drugs, just the essential medicinal list by WHO lists 170 of them, but each of them can be formulated in different strengths, in different dosage forms, injectables or tablets, and so on. And each batch production also varies depending on the capacity of the manufacturer, on the market, and so on. So there are many products available. Anti-cancer drugs, just 127 of them listed by WHO as essential medicine list, and a variety of products, a variety of manufacturers, and so on. Next slide, please. So these anti-cancer drugs, they are in crosshairs for pharmaceutical crime. The reasons are the price, as we are familiar, is very high. There is a presence of gray market rather than that of the legal market, just markets which are not undergoing regulatory scrutiny are available. Cancer epidemiology is increasing, so they are in high demand. There is that of the epidemiological transition in low and middle countries too. So with rising cancer, just the demand is high. And most of the countries in low and middle cap countries, they just lack that of the regulatory capacity. So the anti-cancer drugs are in crosshairs for a possible pharmaceutical crime. Next slide, please. So drugs should fulfill the required quality, safety, and efficacy, and that is set up by different pharmacopoeias. There are 21 of them, like United States Pharmacopoeia, British Pharmacopoeia, International Pharmacopoeia, Indian, Chinese, and so on. Even African countries like Egyptian Pharmacopoeia, South Africa also has its own, and so on. So in this pharmacopoeias, the monograph contains detailed information how to perform just regulatory checks in the laboratory, like identity tests, determining that of the active pharmaceutical ingredient, assay, determining the packaging and labeling, and certain performance characteristics, including dissolution, disintegration, and so on. So this kind of tests have challenges and limits. The tests are high in cost, and that is unaffordable by low and middle income countries. Also, harmonization incentives are there, but still they are not successful. There is no acceptance of one test for every country. So each regulatory authority in its jurisdiction is performing its own tests, even though just regional entities and many harmonization effects are in place are still ongoing. Next slide, please. So just for ascertaining the quality of drugs, we have to be dependent on certain technologies, like the full compound LTs, which require lab analysis for anti-cancer drugs that has to be done in in an engineered way where safety of the analysis is kept. So it needs that of safety hoods and so on. And these are mainly available in high-income countries. Other technologies like scanning technologies, spectrophotometers, Raman or near infrared, the cost is from 6,000 to 60,000, but they rely on libraries. So authentic samples has to be scanned first, and that library has to be built. If that library is variable from manufacturer to manufacturer, so for every manufacturer, we have to have a library. That is the limitations. The other easy-to-use technology, which is highly widely used, is that of the mini lab, supplied by that of the GPHF in Germany, supported by the pharmaceutical giant Merck. But this TLC procedure currently doesn't have monographs or procedures for quality control of anti-cancer drugs. So most of the just regulators, as well as that of healthcare workers in low and middle income countries, they just highly depend on that of visual inspection. But visual inspection depends on change of color or something, a defect which is highly pronounced that we can catch it by eye, but that cannot catch all defects. There are many things which are not observable. Next slide, please. So for anti-cancer drugs, just the chemo part come on. This chemo part is just size of playing card, which contains many reagents that are deposited on it. And once the reagents are deposited on it, once we apply that of the anti-cancer drug just for screening, and just we allow it to run like that of the light chromatography, different color codes are produced. That color code production depends on that of the chemistry of the reagents as well as that of the chemistry drug. So that color is observed visually, and that's how it works. So initially, this chemo part was working for 4 drugs, that of cisplatin, oxaliplatin, methotrexate, and doxorubicin, but currently it is just extended to include mesna, hydroxyurea, and that of calcium leucovorin. Next slide, please. So this is a typical example of how doxorubicin is observed. So this doxorubicin is a reducing agent, and also it has some phenolic groups, as you may see in the structure. So just in lane G here, it reduced Van Dyl, and so that dark spot is observable. In the last lane, that of the L lane, so the chemopad card contains 12 lanes. Just by complexation with that of these phenolic groups with iron, it forms out of this orange color. So such kind of observation is that of the basis for the working principle of that of the chemopart. Next slide, please. So these colors can be observed visually, or they can be taken by mobile phone, and that mobile phone image can be read by that of software developed for that purpose. That is called pad reader. And this image analysis software, which is integrated with that of the pad reader, which is freely available from that of the Google Store or the iOS Store, just can easily identify that of the presence of the API. So the identification sensitivity is at 30%. So almost for all drugs, accurately identifies in Hata 100%, but for oxali and cisplatin, they just the percentage slightly decrease. Overall, just for assessed drugs, its accuracy currently stands at 93%. Next slide, please. So in addition to that of identification, this chemopart helps us also in quantification. And from the calibration curve, it can quantify things in 10 to 20% range. That is a good performance because this is a cheap, low-cost technology, costing $1 to $2, and other just high-cost things, easy to use point-of-care technologies like the Minilab also have such ranges. Just they determine that of the API in the range of 20%. So the quantification capacity with integration with that of the pad reader, mobile phone application, and software integration, this technology is a good one too. Next slide, please. This chemopart, it can be used in a compounding area in the hood. It's small size, it's a size of playing card. Before preparing admixtures, just we can assess the quality of anti-cancer drugs, and that information can be passed to that of the oncologist, and switching of brands, switching of high quality medicines for patient benefit can be in a real situation, a practice. Next slide, please. So this is how the photo images or that of the screenshots from pad reader, where it can identify that of the screen drug. For instance, in the first one on the left side, it's identifying cisplatin, and on the right side, it's identifying doxorubicin formulation. Next slide, please. So just the overall aim is sort of protecting the patient in a real case scenario at point of care at first time. Next slide, please. Yeah, just after screening, if you want follow-up, we have to do HPLC. That is the golden standard. And so the HPLC, we have to perform assays by HPLC and release that of the peak area to the concentration and determines that of the exact API percentage assay value. We are also validating developing methods, and they are transferable. For instance, in the slide, the one that's developed in USC at the University of Notre Dame by Professor Maria Limerman group is capable to be translated, work exactly in the same condition at Addis Ababa University. Next slide, please. So this is a typical example where when a field test out of the chemopart that captures substandard cisplatin. We collected around 20 vials of cisplatin, which are distributed along 5 patches. And when HPLC is performed by taking the sample to that of the USC at the University of Notre Dame laboratory, the concentration is almost half, 54% on average. This is easily detected by that of the chemo part. As you may see, on the left side is the standard 1 milligram per ml concentration, but on the right side, the color intensity minimum, because it's almost half of it. Next slide, please. Yeah, the producer of this substandard cisplatin product, it was also producing other batches, which is captured by that of the Gujarat Medical Service Corp, an Indian regional regulatory body, and it's producing drugs as which their test confirms at 40, lower than that of our reported value, and this company was just fined to not participate with tenders for 3 years. So but unfortunately, our patients at Ethiopia were harmed. That is the sad story. Next slide, please. So as a continuation of that, just with this new project, collecting drugs from 4 countries, we collect drugs from Ethiopia, Kenya, Malawi, and Cameroon in 2023, 2024. And just we package them properly with absorbent material, with proper labeling, controlling temperature in case while transportation by that of DHL, FedEx, and so on, there is any harm where during storage out of the control of us, if there is any degradation and so on. So after sending the samples in such a way, just HPLC analysis was carried out at the University of Notre Dame, and results will be presented in the next slide. So the total samples collected were 251. They belong to 191 lots. Next slide, please. So from this, just 32 failed the HPLC assay out of 191 lots. And please continue. From such 32 failures, visual inspection only catches 3 of them. The other, they are not catched with one. For instance, this one is a change in packaging, in writing, and so on. But if that is not filed as a change to the regulatory body, it is just a fake or falsified products. Here, visual inspection all are clear. There is no observance, but the percentage API is highly variable. One is near 30. It's cyclophosphamide sample. The other is 82. You can imagine the range. So visual inspection only just catches some, but it's not to the extent that we need. Next slide, please. So ideally, just a drug should be manufactured properly by a manufacturer with GMP compliance, and any changes should be reported to the regulatory, and in such a way, just safe, effective anti-cancer drugs produced. But in reality, in low and middle-income countries in sub-Saharan Africa, just chemo products may come from the gray market, where regulatory oversight is non-existent, unreported, no central registry of the products, and so on. Even manufacturers may outsource them, and the labeling may be different. Also, outsourcing or contract manufacturing with the regulatory oversight is the best and allowable. Such and so many factors and just pinpoint before that makes anti-cancer products in crosshairs, makes that of controlling the quality of anti-cancer products at risk. So the results are pinpointed to that direction. Next slide, please. So in our assessment, in the 32 failures, just the factors that are not associated with failed samples in the HPLC outcome includes there is no difference statistically from 4 countries, that of the Kenya, Ethiopia, Malawi, and Cameroon figure. Statistically, it's not significant. those samples just collected from that of high public hospitals versus that of private small community pharmacies, there is no statistical difference. Just regulatory registration alone doesn't bring any difference. There were also just some samples, 59 of them, which were analyzed post-expiry, and the failure rate in expired drugs and as well as that of those not expired is not also different. Although if a drug is expired, it should not be used at all. However, from 50 stated manufacturers on the label, we are not checking exactly the manufacturer. We are relying on that of the label here. We just found that 31 of them, they don't have any failure. But that of the 5 stated manufacturers, they contribute significantly for 27 of them out of the 32. So if there is a mechanism, ideally, that can remove these top substandard falsified manufacturers, just our figure will be reduced to 3% rather than that of the 1 in 6 that of shocking 70% figure. Next slide, please. This is the API by API failure rate. So the dots represent the assay values that were found by that of the HPLC procedure. In between 90 to 110, this one samples are good ones, but above toxicity range, below it is containing API, which is not on that of the pharmacopoeia limit. So for each product, the assay value is summarized in this dot plot. This is for 191 unique lots, which was done from 223 to 224, published in the Lancet Global paper. Next slide, please. This is the continuation of the assay, where additional lots, 203 of them are included from the previous slide, and still the number is increasing from 32 to 76 percentage, slightly increasing to 90%, even though it's not as significant. So still we are observing failure rates in 4 of the countries where samples are collected and analyzed done in USA. Next slide, please. My colleague Atala will continue. Thank you.
Okay, thank you so much, and then Ibrahim. Yeah, let us maybe take a break. So I'm not a pharmaceutical analyst or a chemist, but I'm a frontline healthcare worker. I'm just encountering this in a day-to-day basis because of actually our position in providing healthcare. So maybe consider yourself as maybe just only 1 minute as a frontline healthcare worker, or maybe as a patient. You can imagine what makes this, if this is actually the real scenario, and then if you are actually the one who are actually affected by this problem, it's really devastating. So the impact is not just simply numbers. The impact is is multifaceted. So for example, in real world, the problem in oncology is we don't know, is it because of the substandard falsified medicine? Is it because of the disease progression? So it might be very difficult even to identify whether it's because of medicine failure or because of disease progression. So it's really the impact is really enormous. It really impacts the patient and their outcomes, and then even the frontline healthcare workers will be frustrated because they are investing their time and then energy just to treat and then cure such a group of vulnerable patients. Next slide, please. So what the thing what I mentioned like earlier is because this is not the responsibility of the pharmaceutical analyst or the chemist. Everyone has a responsibility. At least you can do our part. Like you can do just visual inspection or other day-to-day clinical investigations. For example, if the patient not responding to the regimen, we know that that specific regimen is like effective or curing patients more than 90% of the case. But if our patients are not responding, even if we are using the same regimen, so that we have to be suspicious. And then second, there are easy things that can easily identify during the clinical practice. For example, we know that we are giving cytotoxic medicines. They are really, they produce lots of actually untoward or adverse events. Like patient might have nausea and vomiting, patient might lose their hairs, patient might develop infection and the like. But there are times like even patients are really stable, even if we are giving them high dose and then as to the standard the regimen. No infection, no side effect, but the disease is progressing. So this will make us actually like suspicious and then maybe report it to a responsible body to do further quality analysis. Next slide, please. So that's actually like the point that integrated the analysis with the frontline healthcare workers. So when this project was designed initially, it was like it was actually like designed to check the quality of anti-cancer medicines. Then what? Are we going to translate it to the actual clinical practice? That was actually the question, and then that was actually the point when we started like thinking of the chai basket, for example. If the anti-cancer medicines fails, then what will be the backup? And then we started discussing this issue with the clinicians or frontline healthcare workers to design a strategy, at least when such worst case scenarios happened. So you have to have a backup or a solution to continue the care. So that means like really engaging or having a multidisciplinary team approach is really important in fighting against this substandard and falsified medicine issue. And then in addition, the regulators should be like capacitated. They have to be aware. And then I think engaging them and then probably broadening the scope of post-marketing surveillance is really critical. Next slide, please. So I think that one thing I forgot to mention, for example, I mentioned that in 2017-18, cisplatin was the medicine that actually failed the assay. And then that was reported to the regulatory authority. And then the regulatory authority actually do the further regulatory measures. And then when the Aieno and then Professor Maria and Aieno's team do, the analysis again from the same manufacturer, and then actually all the cisplatin batches actually passed. So this sort of like measures will further intensify the the impact. And then the other issue, as I have said, mentioned before, so we can report like adverse drug events. So that adverse event will be taken as a signal to do further quality assessment and the like. So this was actually one of the report, published in Kenya, as per the Kenya's pharmacovigilance database, and then they found out that imatinib among the anti-cancer drugs were actually the one that was like the most reportable one, and then the most suspected medicine for treatment failure. So such actually like reports can be taken by the regulatory authority to do further investigation and then regulatory measure. Next slide, please. And then the other issue I think I forgot to mention was like there were like one patient who took the hematologist prescribed hydroxyurea, but the hydroxyurea was excreted through feces. The intact hydroxyurea was excreted to feces, and then she reported this issue to the hematologist, and then the hematologist reported this issue to the regulatory authority, and then we found out that hydroxyurea was sourced from the gray market. So particularly like the one thing that makes actually like the regulatory intervention very difficult is like those medicines that are sourced from the grey market. It's very difficult because we don't know their source. We don't know, like, who processed that one. Actually, there are lots of factors in the supply chain process, so the intervention might be very difficult. So, in terms of, like, role of professional societies or associations, I think the report produced by Professor Mari and then the team was actually, like, taken to create more awareness among the healthcare professionals and then the society or the community. So particularly, these giant professional associations will have a huge impact in terms of creating awareness among us policymakers, healthcare professionals, and as well as the community. Next slide, please. So Dr. Yoba will continue the study limitations and the conclusion. Thank you so much, everyone.
Yeah, thank you. Actually, thank you, Ainu and Ibrahim, for the excellent talk, and really thank you to all our audience who have stayed up to this point. So I'm gonna just touch a little bit on some of the limitations of this cross-country study that involved 4 countries, notably Ethiopia, Kenya, Cameroon, and Malawi, and also to highlight some of the key recommendations that we think are important to echo to a diverse audience. So the first limitation, at least from this study, is the narrow medicine coverage. So we tested just about 7 to 9 medicine out of 127 of anti-cancer medicine that appear on the or feature on the WHO emergency essential medicine oncology list. And this means that the findings reflect a fraction of the supply chain, and therefore failure rates for untested anti-cancer drugs remain unknown. But again, based on what I knew was presented earlier, we see that as we increase the number of samples, at least between 2024 and 2026, we actually saw the failure rate moving up to about from 17% to 19%, which indicates that the scale of a problem is large. And perhaps we are quite certain that if we survey medicines from other countries, we're gonna see higher failure rates. The second limitation relates to maximum variation sampling, which was actually designed to catch new brands, batches, and lots, and not to estimate population prevalence. As such, the failure rates that we are observing are just a risk signal and not a precise prevalence statistics. And third, the scope of the study, which actually was limited to 4 countries, and as a result, the findings of our study may not be generalizable to all sub-Saharan Africa, as well as other markets. And as a result, the risk profile may differ. But nonetheless, I think it sends a clear signal to all the countries in the continent that this is a potential public health risk, particularly when it comes to assuring the quality of anti-cancer medications in the respective countries in Africa. And the fourth limitation was that of the testing methodology, which was assay-based, and which only could identify active ingredient, which only could measure active ingredients. And as a result, other parameters were not assessed. And this is important because true quality failure rates are likely to be understated, particularly for products passing assay, which may still fail other standards. And last but not the least, the study did not investigate investigate or establish the root causes to these failures, which may include manufacturing failure, cold chain breakdowns in cold chain, falsification, and so on and so forth. As a result, targeted investigation and direct engagement with regulators and manufacturer manufacturers would be needed for upstream change. And I think Atelier did mention how engaging the manufacturer, particularly NAPROT Life Sciences, about the suspicious finding or the confirmed failure of cisplatin in Ethiopia actually led to improvement. So for us as scientists, our public health researchers, our goal, as well as that of everyone, is really to promote patient safety. And we think these results are signals that will warrant should lead to engagement for improvement of quality, as well as the outcome in patient care and treatment. Next slide, please. So to conclude, our study did find that 1 in 6 anti-cancer medicines across the survey countries failed our assay, and this means that patients are receiving chemo drugs that may either be underdose or toxic, and as a result, these products, when they get them, they may not obtain the therapeutic benefit that these products promise to offer. And secondly, the regulatory aspect, which is really important, as was highlighted by Ibrahim, indicating that most of African countries are still at maturity level 3. In short, most of African countries are still at maturity level 1 and 2, and as a result, do not have the capacity to better regulate these products. As a result, there is that urgent need to build regulatory capacity among the remaining countries so that they can move from level 1, level 2 to at least an acceptable level that could enable them execute their regulatory functions so as to safeguard the health of their populations. And the third important thing from our study is the economic cost. So based on the analysis, I think about 383 million US dollars is being wasted annually on poor quality oncology drugs. So this is a problem in the sense that at least from an economic sense, where the budgets of countries are constrained, and the healthcare budgets are constrained, and where most of the costs will have to be supported by household. So this should also be like a call for concern, because it's an important contributor to poverty, and such should not only be viewed as a medical challenge, but also as an economic and developmental challenge. And last but not the least, 3 out of 32 failures were caught by visual inspection. So currently, across many clinical settings, the only reliable, the available tool to detect this, in short, substandard or falsified medication, is visual inspections. But our study has actually unveiled the limits of this. And this warrants the need to develop tools like ChemoPath that could assist clinicians or frontline staff to identify these substandard or falsified medications before they are administered to the patient. Next slide. So there are a couple of recommendations that we want to offer here, which will target 4 categories of stakeholders: frontline clinicians and pharmacists, regulators, government, governance and policy, as well as researchers and global partners. So for the frontline healthcare workers, I think it's absolutely important that in addition to visual inspection, they should consider the possibility of deploying innovative point of care tools such as a ChemoPath. In addition, there is that need for them to immediately escalate suspected products through the established channels, so that action should be taken. And they raise that need to consistently document lot numbers so as to really establish the repeated failures from same source. And I think if you remember when Ayu was talking, I think when we removed like 5 manufacturers from the from our when we did the analysis without 5 manufacturers, we saw that failure rates actually came down to about 3%. So that sort of pharmacovigilance or those sort of actions at clinical settings can actually help to pick out these sort of substandard or falsified medication. So from the regulatory perspective, I think we think there is that need to build HPLC capacity. There is also that need to target the highest risk manufacturing profiles for priority surveillance, mandated sharing of assay results across country, and for countries to be intentional to make progress towards maturity level 3, so that we can actually expand the number of African countries from 9 maybe to an acceptable number, acceptable number, because clearly 9 countries alone cannot provide regulatory needs for the entire continent. So it's actually true that the African Union is in the process of setting up a pan-continental regulatory body, but these are some of the real challenges that will need to be addressed immediately. So from a governance and policy perspective, there is that need to fund quality surveillance, particularly for cancer products. There is a need to establish clear budgets and or budget lines within government budget, so that these activities can be undertaken. Because without these resources, it becomes very difficult to implement quality surveillance for anti-cancer medications, as was previously described. And then there is the need to perform quality testing on all oncology drugs as they enter hospitals, through different mechanisms, from visual inspection, and use of point of care tools, and also lot tracking. And more importantly, develop continental data sharing frameworks, which could enable countries that detect substandard or falsified medications to escalate that, so that other countries are alerted about the situation, so they can take the necessary actions. We had previously how this sort of engaging manufacturers whenever there's a safety signal can actually improve on the quality of the drug. And researchers also have, but the researchers and global partners, they also have some responsibilities. For our group, I think there is the need to expand the chemopath to coverage, to cover more products, and also to assess the quality in additional countries, so that at least we can maybe survey or assess the quality of a broader spectrum of all the 127 anti-cancer medications that feature on the WHO essential medicine list for oncology products. There's also a need, and I think we have done this, particularly work being led by Professor Sachi, to model the health economic impact, so we can build an investment case. I think we have already published a paper on this for 4 cancers, and stating what the economic burden of using substandard and falsified medications can is. So this brings me to the end of the presentation. So I thank you for your kind attention, and we pass over the microphone to our moderator.
Thank you so much, Yauba and Atale, Ibrahim, and I know. Just listen to this and not be struck by how much this is a crisis for African patients. It's actually just incredible. Congratulations on such a seminal study, because I haven't seen studies that are as detailed as this. And so the point you make, that this study may not be generalizable to every African countries because each context and risk factor profiles are going to differ. I think, nevertheless, that we need more studies like this, and it's very likely that this is perhaps the situation facing more countries than not across the African continent. I mean, if the majority of African countries are relying, or most regulators are relying on visual inspections, and this study is telling us 3 out of 32 failures were picked up by visual inspection alone. I mean, that is catastrophic for patients. It reminds me of a visit that I made to Kolebu. It's the major teaching hospital in Accra. Last year in the oncology unit for pediatric treatment for children. And the oncologists there were talking about how they were doubling and tripling the dose and still seeing no effects in the children they were treating. And this really echoes, what you mentioned, that you have situations where, by all measures, clinical measures that you might have, the patient should be responding because the situation is, the conditions are optimal, and yet you see no improvement in the disease. And so it is terrifying, it is horrifying to imagine that on the African continent, and how difficult it is for patients to access treatment, we are wasting $383 million. Just to repeat that, $383 million being wasted on poor quality medicines. I mean, we do need major interventions, some of which you have recommended here, in terms of what needs to be improved on all fronts, right? And what is striking for me, one of the most striking things for me, is the fact that you actually have a small number of manufacturers who are disproportionately responsible for a lot of the quality failures. I don't know what that means or what that tells you. I'd like you to comment on that when we start the questions. And the fact that registration status didn't seem to have an effect on the rate of failure. If I understood correctly, whether a product was registered or not, you still maybe saw similar levels of failure. So that's actually quite striking for me as well. And the fact that you mentioned, I think it was Atale who mentioned that NAPROD's life sciences product is passing quality testing when it's submitted to regulators, and yet, you're seeing the results that you're seeing. So I think things have gone terribly wrong in the way that we are looking at this, and we need some serious and major interventions to really better protect patients. And with that, I'm going to open this flow to questions. But before we go to the questions, normally we do these polls, and today we are doing them differently. So I just posted here 2 questions. They're very simple, multiple choice questions. They're anonymous. If you could maybe just take a look as we have this discussion and, provide your answers to these 2 questions, and we'll get the results and we can share them at a later time. Now, the floor is open. Audience, you may come in, and we can begin. Tebogo Mazibuko, you got the floor first. Please go ahead, introduce yourself, your affiliation, and then ask your question as briefly as possible. We are a little bit out of time. This was a super interesting presentation, and I'm sure many people will have questions. So Tebogo, and then we take Solomon, and then Mujahid, in that order.
Okay, greetings, everyone. truly we are faced with a catastrophe, because after this is the most comprehensive study that I've ever been in a meeting to just sit down and listen. And after this, I'm like, what is happening in our countries? So this, what I have is not a question, but it is like, this is the day that I never thought I would live to see, whereby I would attend a meeting that is addressing the issues that I already have solutions to. Like, I don't even know what to say, because I've written, you see this paper here? These are the issues that are raised, and I'm really happy to say that we have the solution, like at this point. And I really would like to have a conversation with somebody I can just talk to regarding this system that we have built. So we have a solution to what we're talking about today. So that's what I wanted to say. There is a solution already, the system that is running, and we would like to introduce it to the world.
Thank you, Tobogo. Solomon, please, your question.
Thank you very much. I also wanted to thank the researchers on these very important issues. They did a lot, and they showed us what the gaps are in Africa, especially in the areas where they focus their studies. They showed how the policy could improve the cancer medicine supply system in this world. And what made me very interested on this research is it includes from the laboratory to the practice part, so that it is inclusive, and they tried to include the problems from end to end, from laboratory to the practice. This is also one of the strengths of these studies. When we come to one comment from my side, in their study or in their finding, what I heard, if I am not mistaken, there is no difference among countries on the falsified medicines. There is no significant difference among the countries. If this is the case, one of the recommendations from their side is to progress toward this ML3, as only 9 countries in Africa cannot handle the problem in Africa. So if they believe that if there is no difference among the countries, if there is no difference, it results among the countries which have WHO maturity level 3, and for example, Ethiopia has a maturity level 3 country. Kenya is not currently. So if there is study showed there is no significant difference between the 2 countries, what they expect if the number of countries with WHO maturity level increased? One question is this. The next is, what do you recommend?
Can we just, can you hold your question? Because we have quite a few questions. We'll circle back to you, but we'd like to take other people's questions as well, because the time is running out, and we can come back to you. All right.
Thank you.
So let's take Dr. Adol. Please come in with your question, and then Mujahid. We're going to take them in blocks and then let the speakers answer.
Thank you very much, Lenias and the team, for the presentation. Since we're talking about dealing with the menace of counterfeit medicines, and we've seen the magnitude of the problem, because I just thought that the presenters are really just giving us a tip of the iceberg as far as the issue of counterfeit and substandard medicines are concerned, especially in critical areas such as oncology. Now, we talk about detection using laboratory methods, visual inspection and things like that. But I guess for me, being a patient safety expert, I thought the onus lies a lot on the healthcare providers who are dealing with the product, as well as the regulators' ability to actually detect, identify, and come up with solutions based on clinical experiences. So let me explain. So it's one thing for all these drugs to pass across the supply channel and end up with the doctor. But it's one thing for the doctor to realize that a medicine is not working, and for that doctor to take action. And in this case, what we require from healthcare providers is actually a reporting. So we need to find a way of encouraging doctors and healthcare professionals to report what I describe as a lack of efficacy, especially with vital products such as oncology products. The next thing is we talk about maturity of NRS. So the maturity for me in terms of patient safety is actually the ability to collect, identify, characterize and actually communicate patient safety issues, especially with oncology. And then the next one, the onus is also on the manufacturers, right? So the manufacturers, if they don't know what they're doing, if they don't know that the product that they're supplying are ineffective or they're not great, the onus is on them to actually report, because sometimes they don't know. So, I mean, it's a three-pronged approach in terms of the reporting, the analysis, and the communication of the clinical effect of these products.
Okay, thank you. Thank you, Dr. Ado. Mujahid, we'll take your question.
Hello, hello, Ma. Can you hear me?
Yes, we can hear you. Make it brief, okay, if you can.
Oh, okay. Here is just 1 to 2 questions I have. One, I just recommend the presenters of this indispensable presentation of oncology medicine, because is that what is painful in most African countries, more especially Nigeria. You see that many patients were suffering from chemotherapies, but still they are not yet recovered, and due to lack of that substandard and falsified medicine. What I recommend here is that why Africa can't start thinking of manufacturing their own oncology medicine. That is one. Secondly, 95% of the oncology drugs were imported from India, and those importers and exporters, they are not genuinely licensed and authorized by even their Indian country and even to the country that they are going to import it. More especially in African countries, you see that many people were just importing oncology drugs and selling it under unregistered pharmacies and other healthcare professionals. So then three, the chemotherapy part. I think there is need for that chemo part to be available in some African countries so that it helps to reduce the circulation of that substandard and falsified oncology drugs. That's all what I can contribute.
Thank you. Thank you, Mujahid. We'll take one last question, then we bring the speakers to answer them in a group, okay? And the speakers, you can start to think of which one each one of you is going to take. There was Angie something. Anjum, do you still have your question? Your hand has gone down. Okay, we'll go with these questions, and meanwhile, people can also raise their hands and ask their questions. So to you speakers, back to you. So no difference in maturity status of national regulatory authorities in terms of the failure rates, because the expectation is that maybe a more mature regulatory environment should have fewer failure rates, because it's assumed that they have greater competencies in terms of market surveillance and detection of these products. So what is the significance of that in your perspective, in your view? I don't know if I captured the question correctly. And then Dr. Ado's questions on doctors taking more action, being able to report more. I'm trying to paraphrase here very quickly. And can Africa start manufacturing locally? Over to you. You can just take whichever question you want, and we can talk through them.
Yeah, thank you for your comments and conclusions, Dr. Lenias. And also, I want to address the first question by Solomon. Just maturity level 3, if there's no difference by each country, why the maturity recommendation is there? Unfortunately, the maturity level 3 achieved by Ethiopia is very recent. Before the study, just all are on the same phase. They did, any of the 4 countries didn't achieve that of the maturity levels 3. So that may be the factor. However, when regulatory agencies such a maturity level 3 just expected to perform mature regulatory functions, preventing their population from substandard and falsified medicines. I think the timing may be a factor. That's my comment. For other questions, I give to other participants. Thank you.
Can I just jump in and maybe briefly comment on the maturity levels and as well as the question about local manufacture, why Africa can't manufacture their products? I think the question raised by Mujahid from Nigeria. I think with regards to regulatory, with regards to maturity level, I think one of the things we need to perhaps understand is that WHO has like a benchmarking tool that they use to assess the capacity of a regulatory authority. And if that benchmarking tool is actually, if that assessment finds that a regulatory authority meets meets certain some of the indicators there, then they can now rate that regulator, they can now define that the maturity level based on those indicators. So one thing that we need to take into consideration, and this is really a question that I ask, particularly when it comes to effective implementation of the responsibilities of the maturity level or the implementation of the responsibilities of a regulatory authority, irrespective of the maturity level, but more specifically, maturity level 3. Particularly in the context of Africa, we have seen that there are about 127 antimitotic medications on the WHO essential list of medications. And believe me, we need quite a lot of resources. The regulatory authority, even at maturity level 3, will need quite a lot of resources in terms of human resource, in terms of financial resources, in terms of the technology itself, to be able to adequately perform these functions. So even if the rating is good and those resources are not there, the financial resources, the human resources, the technological capacity, then it becomes very difficult for that regulatory authority to execute those statutory functions. So that is one. And then the second thing is, generally speaking, when manufacturers submit a regulatory dossier for approval, I think they usually submit samples, right, which are assessed, and most of the times these samples can be good, they can pass the assay, but what about subsequent samples, right? What about subsequent lots and so on? So we need to like really check how lot-to-lot consistency, regulation of lot-to-lot regulations is happening across those regulatory authorities. So it's really a complex thing. It's really a complex thing, and really I'm kind of excited about the moves that the African Union is taking, particularly pushing towards regulatory harmonization by setting up the African Medicine Agency, which which can actually play this function. Hopefully, they're gonna get the resources, financial resources, human resources, and also technological capacity to be able to perform these things, these functions at the continent. And really, what we are looking for are kind of two maybe regulatory mechanism. One is like the EU style regulatory approach where a product is like reviewed by that agency and then have access to all the markets and then controlled centrally. And secondly, mutual regulations, particularly where maturity, where more mature, where regulatory decisions made by more mature regulatory authorities are actually recognized in other member states. So there is a lot to be done from that front. And then let me just briefly talk about local manufacturing. And this is really like a very good question, and it's really a thematic or a topical issue within the continent. So I think last year there was like the African Health Sovereignty Summit that was held in Kenya, in Ghana, under the leadership of President Mahamat. And after that, I think a couple of conferences or high-level meetings have been held in the continent to actually discuss about health sovereignty. And one of the key things that actually features in this agenda is local manufacturing. How do we get local manufacturing in the continent? There is a lot of political interest and political and diplomatic interest in getting this right. And happy to say that progress has been made for a number of products, including vaccines, antibiotics, and the like. And today, African Union has an inventory of manufacturers. But nonetheless, this aspect of manufacturers for oncology products is still lagging behind. So I think it's absolutely important for the continent as a whole, for countries as a whole, to really take this bold move to set up pharmaceutical companies that can manufacture oncology products. So, but this requires a lot of investment, a lot of capital, and generally speaking, manufacturers or investors will kind of categorize the environment as a high-risk environment, but from a perspective of health sovereignty, continental sovereignty, this is a tough question that we need to find solution to. And I'm really happy that the head of states of the different African countries, they have recognized this, they have made this a priority. And I actually encourage you to be watching. I think there is this conference that is actually coming up, the Conference of Public Health in Africa. And one of the track will be local manufacturing. And we actually encourage maybe researchers or maybe policymakers, investors to submit their abstract so that we can actually begin to get a better view. Over.
Thank you so much. And, you know, like you say, I think one advantage to be mentioned about manufacturing products locally, it's so much easier for our regulators to send inspectors to inspect factories that are within the continent. You know, if you have to send somebody from, I'm sure, from Cape Town to Accra or to Nairobi, it's much easier perhaps than trying to send inspectors to India, to Gujarat, or to another country. So that's another advantage to mention. Lydia and then Richard.
Sorry, can I say something?
Sorry.
Yeah.
One speaker
Yeah.
still wanted to say something. Yes.
Sure. Yeah. Sorry, Lydia, for the interruption. I think from the reflection, what I understand is like, as you all know, the substandard falsified medicine issue is a complex problem. We can't design a single solution, or we can't probably say that there is a problem and then that's the solution. So I think we have to, think of like end-to-end, issues or visibilities. Like the manufacturers has to have, execute their own responsibility, supply chain actors, the end users, and patients themselves as well. So even to reflect on the one one comment, I forgot his name, regarding the role of healthcare professionals. Like it's really like, it's not as simple as it is. For example, there's a patient, you don't have any backup, and then you are reporting, and then you found out that this medicine is substandard falsified. So how can you tell the patient, like they are waiting you to bring some solution, that the solution is affected because of this issue. So it's very difficult. You are right. We have to capacitate healthcare providers, even if it might be very difficult to identify whether it's the disease is progressing because of treatment resistance or it is because of substandard falsified medicine issue. So, but I think reporting is by far better than just keeping quiet and then seeing patients dying in front of healthcare providers. Thank you so much.
Thank you, Atale. And you're right. I mean, the solutions that are needed are so much more complex. They are super complex. We need to strengthen market surveillance. It's not happening. Adverse events reporting is not happening. Testing is not happening. So the breadth and scope of what's needed that's not happening is so enormous. And I think one piece that could be very helpful, and I think you did a great job, our panelists, in terms of recommending the simple things that do not need billions of dollars in investment in order to execute them and still be able to protect patients. For instance, countries being very intentional in terms of, how they're interacting with counterpart regulatory authorities where these medicines that are harmful are coming from. I think that's a real potential solution that if it was executed at the level of the African Medicines Agency, from a centralized perspective and then trickling down to national level, that could be quite impactful. So you don't end up with a situation like the one that you mentioned of a manufacturer who had been banned by Gujarat Medical Services Corps in India, and yet they were responsible for a number of failures because they were still able to sell their bad products to Africa, and yet in India, where they make, they've been banned. So those are simple things that we can do, that people like that should not be allowed to bring their product into African markets. And then, there are simple things like being able to make the communities, our communities in African countries are aware, because that's one piece that we see at Medicines for Africa being really a very neglected part of the solutions. Because, if there's awareness, a level of awareness amongst communities, amongst patients, I think that can also serve as a layer of protection in terms of people being able to question and think, perhaps something is not right about this situation where the patient is being treated and nothing is getting better. So just to add to your points, Lydia, please, your question.
Thank you, Lenias. It was actually an additional point that I wanted to make. So first and foremost, thank you to all the presenters for this really comprehensive presentation that you have given us. So when it came to Solomon's question, I'm so happy that Ayeno mentioned that Ethiopia is fairly new in terms of attaining its maturity level 3, status in terms of regulatory capacity. And the point that I wanted to raise is, I think if we were to repeat this study, include more countries, and actually have more of our ML3 countries, I am willing to bet that the results will be different. And I say this as someone who has practiced in an unregulated market and who has benefited quite a lot from both regulatory intelligence and cross-border collaboration with colleagues who are based in South Africa and Zimbabwe. We cannot really neglect the importance of having in place mature regulatory systems in terms of averting and solving some of these crises. So I think we're a bit over time, but it would be so lovely to discuss this, in further detail in future forums. Thank you.
Absolutely. Do we have more questions? So, because our audience has been very, very patient, and of course, this was a really engaging, informative, and very educational presentation in terms of the scope of of the analysis that you have brought to this, and I think definitely one thing we can say is that we have a great need for more studies like this. You gave a number of the scale, the estimated scale, that you think maybe is the number that you can put on it, which was 14%. And I think what would be good is to really get a grasp of the magnitude of this, because time and time again, a lot of the speakers, pointed out how much this is likely to be an underestimation, in terms of the failure rates, and so forth. So what would be good is to be able to really understand how big is this problem and what does it mean, right? Because like Atale said, it's not just numbers, right? These are people. I mean, it takes me back to my time in Accra. I spent a day with moms and dads with their kids there with big souls, and it really shows you these are people, these are families. It's not just numbers or statistics. And so our efforts, or the efforts that our regulators and everybody else who should be involved in this needs to put that front and center. It's about the patient. It's about people, right? And I think that has been missing in the way that we talk about this problem in African communities. We talk about everything else. We talk about regulators. We talk about, yes, we need all of these things to be part of the solution, but we must not forget the communities, the patients themselves, and the key role that they play in this, because they are part of the solution. So let's as we go forward, I think this is something to think about, expanding the scope of this study so that we have a better understanding of just what it means in terms of people, real lives affected, $383 million wasted on poor quality medicines that do not treat, that probably leads to conditions getting worse. What does that mean in terms of children who have died, or families, women who are dying from a preventable disease like cervical cancer, which does not kill women anywhere else in the world except here on the African continent? So I think, this is a very important conversation, and I would like to invite the speakers back, because we barely scratched the surface of the issues, of the many, many issues that you brought up here. And so we need to do more. We need to think more about the things that we can do now. Yes, we need investment in laboratory testing capacity and effective methods and so forth. But what are the things that we can do right now that can really help protect people? And how many people in these $383 million wasted? What does it mean in terms of people? So I think it's been very helpful. Thank you. This was a very powerful study. For me, I learned a lot. A lot of our youth community are here. I see Radagao on the line. I see many of you. And I think one of the things we're trying to do at Medicines for Africa is to open your eyes to the scale and magnitude of the problems that we have in our communities. And these are our sisters, our nieces, our kids, and so forth, so that you can see where you can come in as a pharmacist, as a young pharmacist coming out of graduate school. There is so much that you can do in your own community. And so this is what I would like to encourage you to think about, that you can be agents of change in your own communities. And, there are ways that we can work with people like the team that presented here. How can we, people in the communities, be a part of this? How can we be a part of the solution? I think that's a really important question that we need to ponder and think about, and really, as we go forward, think about how we include patients, make them front and center of the solutions that we come up with. So I would like to thank the speakers for a very powerful study, for highlighting the breadth and scale of the problem, and that we are barely scratching the surface in terms of the impact this has on people, on life. So thank you for that. Let's think about the future, how we can expand on that and bring better understanding, bring better solutions, working with all of the key stakeholders that need to be a part of this solution, including people in our communities. And I would like to thank our audience for hanging on, and for staying and becoming part of this conversation. We are so grateful. This is so important for all of us, for every single one of us here. I think we've all been impacted in some way. We can all say that. And so it's our problem as people in communities. It's our problem as regulators. It's our problems as ministries of health, as people, custodians of policy. It's the problem of the African Medicines Agency, and we need everyone to come up with solutions that work. So thank you very much to everyone for this evening. Speakers, maybe 30 seconds each. Last reflections before we close.
Yeah, just for the chemopad availability, it is a product under research, but everybody can reach the research team, email us, so we can collaborate. Thank you.
Dr. Yauba, your turn.
Thank you very much. And I really like the way you put it, Lenias. We have a very huge responsibility, not only from the perspective of assuring quality access to quality anti-cancer medications, but the cancer ecosystem in the continent needs a drastic transformation in terms of diagnosis, in terms of awareness, in terms of patient support, in terms of funding, and so on. So it's a very, very vast area, and that is really one of the reasons why I appreciate what we are doing. What we need at this point, the starting point, the low-hanging fruit, a simple solution, is awareness raising, and I think that is what we are doing, and we really encourage you to keep on sounding the words so that they can get to the ears that can take the decisions, the regulators, the policymakers, the manufacturers, the patient groups, the supply chain distributors, and the like. So we need also to bring the civil society on board, because as I can tell, they are a critical player that can actually help us solve this multidimensional problem. Thank you.
Thank you, Dr. Yauba. Ayanel, please. Go ahead.
Atalay, I think I already saw something.
Oh, your hand is still up. I see. Sorry. I was looking at your hand. Atalay, do you have some last remarks?
Okay, yeah, thank you so much, Lenias, for the opportunity. I think this is a very good platform to bring more solutions to the continent. So let us work as a team and then at least contribute. I mean, our part. That's what I want to say. So have a good day. Thank you, everyone.
Thank you. Thank you so much. Ibrahim, you have any last remarks?
I thank you very much for giving us an opportunity to present our results. Whenever we have projects like this, one of our aims is to do the dissemination of the results to different stakeholders. And you just provided that, and we encourage you to continue such a wonderful job. Thank you.
Thank you so much. So thank you to our speakers, our excellent audience, our youth, many of whom are in the room. Thank you so much for today, and we look forward to continuing the conversation. And speakers, we hope to see you back so we can really delve a little deeper into the many issues you raised today. Thank you, and have a good evening, everyone.